In this video, we are going to talk about cellular respiration. We'll start off with an overview about the process of cellular respiration. The chemical reaction for cell respiration is C6H12O6 plus 6O2 yields or makes 6CO2 plus 6H2O plus ATP. And ATP is our energy molecule. So this means that with 1 molecule of glucose and 6 molecules of oxygen, cells can make 6 molecules of carbon dioxide, 6 molecules of water, and ATP energy. What you might notice about this reaction is that it looks like the reverse or opposite of photosynthesis, even though our form of energy that we have is different. The location of cell respiration is the mitochondria. So let's draw a diagram of the mitochondria so that when we talk about where these specific processes in cell respiration are taking place, we know what we mean. So mitochondria are kind of a bean-shaped organelle. We have an outer membrane and an inner membrane. And then on the inside is this squiggly thing. That's called the matrix. We could call it the matrix of the mitochondria or the mitochondrial matrix. So that is what a mitochondria looks like. Cell respiration occurs in 3 distinct steps. The first one is called glycolysis. The second one is called the Krebs cycle with a capital K because it's named after a person. This is also sometimes called the citric acid cycle. And the third one is called the electron transport chain, which from now on we will abbreviate ETC. Just like with photosynthesis, in order for respiration to occur, we also need some electron carriers to be able to move electrons where they need to go after they're broken off what they were attached to before. We also need, one of them is called NAD, which turns into NADH when it has its electron, and FADH, which becomes FADH2 when it is carrying an electron. So once again, these are our electron carriers, and they help move electrons to where they need to be during respiration because we can't just have electrons floating around wherever it is they want to be in a cell. It's not going to be a good time for anyone. Next, we're going to go through each of the steps of cell respiration and talk about what happens, where it happens, what's used, and what's made. Starting with glycolysis because it happens first. Oops, I am writing the wrong thing. Hang on. Start with what happens. My apologies. During glycolysis, one molecule of glucose is split into 2 molecules of pyruvate. Pyruvate is sometimes called pyruvic acid, but they both mean the same thing. So essentially what we're doing is cutting glucose in half. And during that process, we are going to have to make 4 ATP and use 2, which means we have a net of 2 ATP. Because this whole point of respiration is to make energy. So we're going to keep track of how much we're making at each of these steps. So during glycolysis, we split glucose in half. We make 4 ATP, but we have to use 2 to help break some of these bonds. So we're only really gaining 2 ATP. The location of this process is in the cytoplasm. So glucose is coming into the cell. Maybe it's just left a chloroplast if you are a plant cell, or maybe with animal cells, it has been consumed and carried to the cell via blood. We are going to use glucose and we are going to make 2 pyruvate. We have our 2 ATP, of course, and we are going to make NADH, because when we split the glucose, we will have a couple of extra electrons that need to be attached to something. So we'll make a little NADH, and that is glycolysis. Moving on to the Krebs cycle. Starting with what happens during the Krebs cycle. We are going to finish breaking down glucose. So we started the breakdown during glycolysis. We're going to finish it during the Krebs cycle. The location of this process is in the matrix of the mitochondria. So our pyruvates from the cytoplasm have made their way to a mitochondria at this point. We are going to use pyruvate, NAD, and FADH, and we are going to make CO2, which will be released, 4 ATP, NADH, and some FADH2. So all of the hydrogen electrons from the glucose are either attached to NAD or FADH2 at this point. The key thing about the Krebs cycle, or a key thing, is that it does require oxygen. Last, after glycolysis and after the Krebs cycle, we have ETC, or electron transport chain. What happens? We are going to use NADH and FADH2 to turn ADP, which is our adenosine diphosphate, into ATP. And then ATP will be used for lots of different things. So it could be used for cell transport. It could be used for cell signaling. Lots and lots of different options there. This process happens in the inner membrane of the mitochondria. So our electrons are going to be pumped back and forth across the inner membrane to harness the energy so we can get some ATP. We are going to use oxygen, NADH, and FADH2, and we are going to make H2O and about 32 ATP. So this is our big ATP generating thing. And then, as a side note, we'll also make NAD and FADH. And those will be sent back to the Krebs cycle and glycolysis to pick up more electrons. The electron for ETC, oxygen is the final electron acceptor. So we're breaking these electrons off of the glucose and they get shipped around and shipped around and shipped around, finally get attached to oxygen, and that's where we get our H2O from. One final note is that the purpose of cell respiration is to make ATP. Water and carbon dioxide are a byproduct. So our goal is to make ATP, happen to make water and CO2 as well. And that is all I have for you for cell respiration for now. We'll pick up with part 2 next time.